Okay, let's finish up heredity. So Mendel's two laws explain the inheritance in terms of genes that are passed along from generation to generation. Basically, the rules of probability. They're valid for sexually reproducing organisms, so not things that we clone. But Mendel's laws stop short of explaining some of the patterns of inheritance that we see. For most sexually reproducing organisms, in fact, Mendel's rules cannot account for some of the relatively rare patterns that we see. Most often, the patterns of inheritance are a little bit more simple than big A, little a, and just one trait. We are not, in fact, peas, so that is a problem. One of the things we see is something called incomplete dominance. So this would be hybrids have an appearance in between. So there's a protein for red, and they look white if that protein isn't there. So in fact, the in-between, or the heterozygous, is going to look pink. So incomplete. There's not enough protein to make red, so it therefore looks pink. You would see this in something like a red flower. So if there's no big R present, you get white. But in between, you have enough to make a little bit of a pigment, but not enough to make red pigment. And that is called incomplete dominance. And therefore, your phenotype and your genotype are going to look a little bit different. So you're going to get a different phenotype for your big R, a different phenotype for your heterozygous, and a different one for little r. Another example of this is hypercholesterolemia. I cannot say it. I just can't. Basically, it's a trait that is an example of incomplete dominance, and it's characterized by high levels of cholesterol in your blood. Essentially, if you're heterozygous, you have blood cholesterol about twice the normal. Homozygous has about 5 of the normal level of blood cholesterol and may have heart attacks as early as age 2 due to high cholesterol. So, right, 2-year-olds aren't eating badly. They have a genetic disease. And it looks like this. This is your cholesterol. This is a normal cell. What would happen? This is if you had the mild disease, and if the disease was severe, you just really wouldn't be able to have any receptors, and it would cause you to have a heart attack at a very young age. Another example of variation is called what's codominant, or also multiple alleles. Sometimes there's a third allele. So in the case of blood type, you can be A, you can be O, you can be B, you can be AB. So there's 4 options in terms of your blood type. The ABO blood groups in humans involve 3 alleles. A person's blood type might, again, be A, B, AB, or O. And the letters refer to the carbohydrates designated as AB that may be found on the surface of the red blood cells. And this has really revolutionized people's ability to have surgery and survive surgery, because now you can have your blood, you can be transferred with your own blood, and it will not reject it, and therefore you will not die during surgery, which we're all excited about. This is essentially what happens. You can be AA or AO and still express as an A. You can be BB or BO. You can be AB, and you can just be O. And we'll talk about how those interact when we start doing some genetic problems. And essentially, this is what it looks like when your blood gets typed. Matching compatible blood groups is critical for safe blood transfusions. If your donor blood cell have a carbohydrate that is foreign to the recipient, then the immune system will produce an antibody that binds to that and causing the donor blood to clump together, and it will potentially kill you. So you really need a blood type transfusion by someone who has the same blood as you, or someone who has a compatible blood type. So there's A, B, and I, which we call O because it's neither A or B. And you inherit one of these from each of your parents. Because there are 3 alleles involved, there are 6 possible genotypes, and we'll look at that in a minute. Both the A and B alleles are dominant to the I. So if you have one of these A and one of these I, you're going to express A. If you have a B and an I, you're going to express B. People of genotype AB make both the carbohydrates. In other words, A and B are codominant. They are both expressing at the same time, meaning that your heterozygous is expressing both of them. Pleiotropy is when one gene influences several characters. So in sickle cell anemia, in sickle cell disease, it exhibits pleiotropy, results in an abnormal hemoglobin protein, and causes a disc-shaped red blood cell to deform into a sickle shape. In most cases, only people who are homozygous for sickle cell have symptoms of sickle cell, but if you are heterozygous for sickle cell, they recommend that you not do things like climb Everest or Kilimanjaro. They will actually test your blood to make sure that you are not a carrier, so that you can perform these activities that require a great deal of red blood cells. So again, this is what your blood would look like if you had sickle cell. Polygenic inheritance. This is the additive effect of 2 or more genes on a single phenotypic character. So it's basically the logical opposite of pleiotropy, in which one gene does several things. This is, several things are, it takes multiple genes for the effect. There's evidence that height in people is controlled by several genes that are inherited separately. It's probably affected by a great number of genes, but here's what it would look like simply. So this would just be all the genes that affect your height, potentially your skin color. It's just a variation of the extreme tall versus the very tall and the very short. You're mostly going to be along this middle axis in terms of height. So it's multiple genes interacting to make your height. Epigenetics. So epigenetics is fascinating. I am fascinated by epigenetics. This is really how your chromosomes are wound, how your DNA is actually wound through your histones, and how that ravels and unravels, and it gets replicated and translated and transcribed. That happens with your histones, and sometimes that can be just your environment and the chemicals you're exposed to, the UV light you're exposed to. So this is the age-old nature versus nurture. What happens when you have identical twins, or why are brothers and sisters so different, if it's all genetic or if it's all nature versus nurture. So this is a huge controversy. Here's a set of identical twins with different diseases. Only genetic influences are inherited and not passed on to the next generation, but the way your DNA is potentially wound around these histones can be affected by the environment. So how tightly or loosely they are wound and then wound into your chromatin and your chromosome can be denatured by things like extreme exposure to UV light, different chemicals, different environmental exposure, things that you eat. And this then will be, this chromosome is inherited, so the way that winds around will actually be inherited by generations to come. For example, components of chromosomes can be chemically modified by adding or removing groups. Over a lifetime, the environment plays a role in these changes, which may explain how identical twins suffer from a genetically based disease where the other twin does not. So we see this occasionally, it happens, and it happens mostly because of the environment the other twin was in. Their genetics are the same, but they were exposed to different chemicals. Epigenetic modifications are changes in the gene activity, so they are, again, transcribed and translated, because here you have them, this area before was not exposed, so it was not able to be transcribed and then translated. But if you loosen it, then the area is exposed, and that gene can potentially be transcribed or translated. This goes along with the chromosome theory of inheritance, which states that genes are located at specific positions, and the behavior of those chromosomes during meiosis accounts for inheritance patterns. So it is chromosomes that undergo segregation and independent assortment, and they account for some of Mendel's laws. So again, we have meiosis. This is your gametes. Your gametes are your egg and your sperm. So this is the process by which you have 2 copies, and now you only have 1 copy because that is your egg and your sperm. When the egg meets the sperm, now you have 2 copies. So these are egg and sperm. They only have 1 copy. When they come together for fertilization, now you have 2 copies, and then those can be divided and sorted to make the full actual human or plant or whatever happens. So this follows the law of independent assortment, and it also follows the law of segregation. So this is how we get those individual chromosomes. So if this is you, right, these are your parents, who one of their genes, one of the other genes, this is the dad because it's a Y. Oh, no, they're a little big Y, little Y. So this is mom and dad. They made you. Now, when you go through meiosis, these are the possibilities of the gametes you could potentially give your child. And that